we'll introduce Andrea. Andrea's going to talk straight through, and then we'll do the questions at the end. So make a note if you have any questions as we go along. Go ahead.
Very,
Take care. Take it off. Take it away.
very good. Welcome, everybody. And if you had a chance to read the summary of today's program, can you hear me okay, by the way? Can you hear me? Okay. If you read the summary of today's program, you know that Andrea and Don Stierle are research professors at the University of Montana. They're doing extraordinary original research in the Berkeley pit, which is what Andrea is going to present on today, and then take your questions. What you may not know is that they have been featured in national media. And so they've been featured on Radio Lab on NPR. They've been featured in an Atlantic Monthly article. So they have some national coverage. They're also highly creative people. Don, for example, is a very fine potter. And Andrea is a skilled painter. I love her paintings. And then you may not know that they're incredibly engaged with the community. They give back a great deal, and they're particularly committed to working with younger Montanans on gaining science literacy and putting that to work in the world. So please join me in welcoming my good friends, Andrea and Don Stierle.
Good morning. It's a real pleasure to be here. And can you hear me?
Up a little.
No.
Up a little.
You gotta tell her no.
We don't have a good microphone on this computer, so how is that?
That's better.
Okay. I'll try to keep it strong. Okay, here we go. Well, hang on. Stop share. I just lost my PowerPoint. Hang on. I'll be 1 second. I lost my PowerPoint. Okay. I found it again. So what I'm going to be talking about today has to do with an extremely beautiful site in southwestern Montana, the Berkeley Pit. And I think anybody who lives in Montana is aware of this iconic relic of the mining heyday of Butte, Montana. But what you may not know is this is also ground zero for some very interesting biological chemistry. Now, geologists and geochemists have spent years exploring the quality of the water in it as people have mined the overburden, looking for copper, manganese, magnesium, and a number of other ores that have been used for industrial purposes. If we actually look at the ore that's come out of the Berkeley pit, the copper, helped to electrify about 30% of this nation. So during World War II, Butte played a huge strategic role. And the history of that role is really quite fascinating, but that's not what we're here to talk about today. So as soon as I can find my stupid little arrows. Yeah, everybody can see my slides, is that correct? Thumbs up if you can see my slides. Thank you. Now, what I'm really going to be talking about today is what I like to call the world's oldest profession. Now, anybody who knows anything about Butte knows it has a strong connection to the world's oldest profession. And I'm not talking about this young lady. I'm talking about drug discovery, because drugs are found in an amazing array of places. And I would contend one of the earliest pursuits of humans was looking for compounds that could help us deal with a variety of different illnesses. Because no matter how modern we think our illnesses are, diseases like leukemia, various kinds of cancers, viral diseases, bacterial diseases, fungal diseases have plagued humans and other animals as far back as we know. And many cultures have huge pharmacopoeias that talk about different types of plants, different fungi, different animal bodies that have been used to help treat diseases. And so Don and I play a role in this amazing lore of trying to find drugs, and we often look for them in some really odd places. Some might contend the Berkeley Pit is one of those odd places, but hopefully in the next few minutes, I'll convince you that it's a most rational place to look for new drugs. So, what Don and I really are natural product chemists. So, we are trained organic chemists with a medicinal background who look at the natural world for compounds that produce chemicals that might be used to ameliorate symptoms of a number of different diseases or to simply make us feel better. They don't have to treat a disease. So, many of our favorite natural products include this lovely chemical, which I'm imbibing right now. This is caffeine, a compound found in coffee, to a lesser degree in teas and chocolates, although there's a closely related alkaloid that's more prevalent in those. And this is truly my drug of choice. I take care of caffeine just about every morning, um, and it takes care of me. If we look at the next plant, we're looking at a tobacco plant. And tobacco produces another alkaloid. This alkaloid is called nicotine. And nicotine is a bitter principle that in the human body can actually act as a stimulant. Um, why do coffee plants and tobacco plants produce these alkaloids? We don't really know. We think they're bitter if you taste them, so the plants may produce them as anti-feeding agents. So if an insect bites into that plant or a deer bites into a tobacco plant, the bitter flavor may repel them. But that's just a supposition. But as natural products chemists, we don't ask why the plant makes the chemical. We ask, what can we do with that chemical to help the rest of us? This next lovely little character is a penicillium, a fungus that's near and dear to our heart, and of course produces a whole panoply of beautiful molecules that have strong antibiotic properties, including the penicillins. This last plant is particularly dear to my heart. It's the Northwest Pacific yew plant, and it produces a chemical called Taxol, which we now have to call paclitaxel because Bristol Myers Squibb patented the name. And I have to now send them $10 because I called it Taxol. That's how patent law works. But anyway, this amazing plant. Did I get a question? This amazing plant is very useful against breast and ovarian cancer, as well as a number of other diseases for which it's being explored. But Don and I didn't start out in the Berkeley Pit. When we were first married and moved to Butte, Montana, and I decided to get my doctorate in the same field that Don was studying, we were looking at a number of different places around the world to do our studies. And we primarily looked at microorganisms because microbes are a renewable resource. If we go down to Bermuda, where we spent 2 summers, and simply collect sponges or collect tunicates or coral, we are actually affecting the reef systems in an inappropriate way. Some of our colleagues would actually go down to reef ecosystems and dynamite out the reef so that they could do broad scale collections of organisms. And you can imagine when they went back the following year to recollect an organism, they found that the reef had been damaged, and in some cases, the organisms had been completely decimated. So we hypothesized that within some of those organisms were actually microorganisms, endosymbionts, that might be responsible for some of the wonderful chemistry that people thought sponges were producing. So when we went to Bermuda, we spent 2 of our summers there. And indeed, we found an amazing compound, which we call Big Pink 1. We're very creative in our naming because it came out of a pink sponge called Darwinia rosacea that was harboring a pink bacterium that's one of the marine ultramonases. This particular microbe produced a compound that is very highly antibiotic, particularly against Staph aureus. But when tested, it turned out to be a very potent inhibitor of reverse transcriptase 1 that's part of the HIV virus. So the National Institutes of Health began to study this compound. Unfortunately, we didn't have enough funding to pursue it because you have to pay to play. But a fascinating compound, part of a fascinating microbe. Um, we then moved to Glacier because we decided to look at a different watery ecosystem. Glacier is about our favorite place on Earth, so what better place to study? Glacier is the home of the Northwest Pacific yew tree in Montana. There are trees there that are about 300 years old. And when we first met a yew tree there on Trail of the Cedars, there was a tree that was about 5 feet tall, a little shorter than I am. And the ranger told us the tree was 320 years old. And 10 years later, we visited that same tree and it was about 5 feet tall. And now it's 330 years old. So they're very slow growing, but they harbor this amazing chemical. Now, when we started looking at Taxol, that was back in the late 1880s. I'm that old. The late 1980s, early 1990s, about the time that Taxol was coming into the press as a potential cure for cancers, particularly for women who had breast or ovarian cancers that were refractory. So a refractory cancer um, is one that has failed 3 chemotherapy trials. The woman has undergone usually surgery and often radiation, and her tumor is not responding in any way. And Taxol was showing about 30% efficacy in such cases. The problem was most of our yew trees had been extirpated on this continent and burned as slash. They weren't considered desirable wood. They grew as understory under hemlock trees and spruce trees that were being harvested by logging interests. And then the yew trees were simply burned as a nuisance. So the supply issue was a huge problem. So what we did was we looked in the bark of the yew tree to find if there was a fungus that might also have a symbiotic role in the production of Taxol. And what we actually found was in the bark of the yew tree, there is a fungus that grows that produces Taxol without any help from the tree. The tree produces Taxol without any help from the fungus. So this was the second example of this type of co-production of a very complex molecule shown here by both a higher plant and a fungus. And I was able to publish this in Science magazine, and the fungus was named after me, Taxomyces andreana. So I have a fungus named after me. I don't have any children, so I can't name children or grandchildren after Don or I, so I get a fungus. Now, after the Taxol work, we ran afoul of Bristol Myers Squibb, who owned the whole patenting rights to Taxol, and they did not want a fungus on the scene that might produce Taxol for much less money than what they were pursuing. So they ultimately acquired the rights to our fungus, and as far as we know, they destroyed it. And we had absolutely no say in the matter. It happened at the university level. So at that point, we realized in 1995, we needed to turn our sights in another direction to an area that perhaps no one else was interested in exploring. And that actually happened to be a site in our own backyard. Don and I were faculty at Montana Tech in lovely Butte, Montana, where we moved about a month after getting married in 1980. So moving to Butte was actually part of our honeymoon. We went to Mexico first. I got dysentery, and then we moved to Butte. And we fell in love with the place. We really did. It's a fascinating town, but it also happens to have something very few towns in Montana had at the time, and that is a large pit filling with toxic water. And what made the water toxic was a very high concentration of a number of different metals, many of them what are called redox metals, highly reactive oxidizing metals, that was slowly filling the Berkeley pit. This is about what the pit looked like on your upper left. When we first moved to Butte in 1980, it was still a dry, active, open pit. People were still mining in it. In 1982, the pit was shut down. Uh, Company members illegally snuck down to the 3,000 foot level on Earth Day in 1982 and illegally turned off the pumps that kept the pit dewatered. They were mandated to dewater it in perpetuity, and perpetuity lasted, I think it was about a month. So perpetuity means different things to different people. And so the water slowly started rebounding in the pit. And now there's about 80 billion gallons of water currently in the Berkeley pit. Up until last year, the water was primarily dominated by an iron chemistry. And now over the years, they've been dumping lye, different types of bases into the pit. And now I believe it's more of an aluminum dominated chemistry. So that chemistry is changing. It's a very dynamic ecosystem. What was particularly notable about the pit was the water had a pH of about 2.5, which is quite acidic indeed. And that kept the water was constantly dissolving the overburden. So it was actually increasing the concentration of minerals in the water rather than allowing it to slowly go back to a more natural state. So this is the profile of the pit set in bedrock. It infiltrates what used to be a butte in the eastern side of Butte, Montana. So Butte gets its name from these sort of bump-like hills that dot the countryside. The pit itself is about 1,000 feet deep, and the water level is currently just below what's called the critical level. And when the water hits the critical level, that water is going to start leaking into the surrounding alluvium and will actually then start infiltrating into some of the groundwater. At this point, it's sitting in bedrock, so it's fairly well contained. But Montana Resources, which in essence operates the pit, controls the pit, they have a number of wells all around the pit to keep on testing groundwater to make sure that infiltration has not yet occurred. So a dynamic place to work, but what interested us was the water in the pit. So the Montana Bureau of Mines and Geology gave us a water sample in July of 1995, and that water actually contained a number of fungi. Now, most of our microbiology friends told us you will never find anything interesting in the pit because it's just too acidic and too rich in metals. So of course, it's the role of a scientist to prove other scientists wrong. It's kind of a fun thing we like to do with each other. But what we found were a number of different fungi. And since 1995, Don and I have explored the secondary metabolism of a number of these organisms, and in particular, the organisms that are listed on the left. Some of these have proven to produce some amazing compounds, and some just produce interesting compounds. But to us and to the students who have worked with us, because we primarily worked with undergraduates at Montana Tech and undergraduates here at the University of Montana, where we moved about 10 years ago. So these were our newest collaborators. They don't say much, but they give so much, often their lives, to our work. Now, I'm going to explain some concepts because I'm going to get into a little bit of the science behind what we do. a lot of what we're interested in and what anybody who is looking for new drugs is interested in are targeting specific enzyme pathways. Now, all life forms have hundreds of thousands, if not millions, of chemical reactions going on in their bodies every second. And what these reactions are a number of different proteins interacting with each other to either break down chemicals or to produce new chemicals. These reactions are facilitated by specific proteins called enzymes. And if we know that one of those pathways is associated with a disease, a runaway pathway, we may try to inactivate one of those enzymes, we may try to inhibit it, or we may find that if we can enhance that reaction, we can somehow improve human health. And so we're either going to be trying to inhibit or activate enzymes. Second concept, I may mention apoptosis. This is a programmed cell death. Cells die. Every cell in your body is destined to die at some point. And if they do it in a very programmed way, and when I say that, things are actually packaged neatly so that they can be reused and recovered. Apoptosis is an important process that allows us to kill tumor cells. Every cell in your body, if it chose not to die, would basically become a runaway cancer. So cells that have forgotten how to die are cancer cells. So we may try to upregulate apoptosis and enhance it when we kill cancer cells. And we may try to downregulate apoptosis when it starts killing brain cells, say after a stroke. So apoptosis can be desirable and it can be detrimental. Inflammation, I think we're all familiar with inflammation. It's an important part of the body's immune response. It's how our body responds to pathogens and all kinds of dangers. We'll have names like PAMPs and DAMPs. These are pathogen associated molecular pattern and danger associated molecular patterns. If these enter a cell, a whole slew of chemical reactions start, and they are designed to respond to these injuries. Runaway inflammation, however, can lead to autoimmune issues and cancer. And so there's a particular enzyme we're interested in called caspase 1 that we're going to try to inhibit. Many of us now, as we're following the coronavirus, we start hearing about cytokine storms, and that a lot of the damage from coronavirus comes from our body's own runaway autoimmune response. And so it is often our role, and that's why steroids are given in a number of cases, because they will downplay the immune system, keep it from starting to damage. Reactive oxygen species, an important concept in all aspects of human health. The body is constantly producing compounds that are highly oxidizing. These can damage DNA molecules, which can lead to cancer and various mutations. And this is why we take antioxidants, is to circumvent runaway oxidation. When you take vitamin E, when you take vitamin C, you are trying to reduce some of these oxidizing events. This is a great one, epithelial mesenchymal transition. This is my favorite term for the last 5 years. EMT, it's where we take orderly cells that grow in a very controlled epithelium, so basically like the surface of your skin, and dissolves them and they turn into a slurry. So think of Slurpee. The cells become disconnected, they become mobilized, and EMT is an extremely important component of metastatic spread. One of the enzymes that's associated with that we're going to look at is matrix metalloproteinase 3, which we're trying to inhibit. And our last two terms are MRSA and MDRSA. Anybody who pays any attention to the field of antibiotics knows about methicillin-resistant Staph aureus and MDR stands for multi-drug resistant Staph aureus. These are a huge threat worldwide. MDR SAs have been considered by the World Health Organization and the CDC to be part of what they call the escape pathogens, the 6 most dangerous pathogens in the world. And we just happen to have discovered a brand new class of antibiotics from Berkeley pit microbes that target MDRSAs in a pretty amazing fashion. So how do we do what we do? First of all, we have to isolate the organisms in pure culture. Each organism is then grown in a small pilot study, often in as many as 20 different fermentation conditions. When we were at Montana Tech, I would often have 15 undergraduates working with us, and we could have those students doing all of those fermentations. Wonderful human power there. They extract the organic and aqueous solubles. That basically just means you take that slurry and you pour chloroform in it, which is not water soluble, and that chloroform will pull the organic solubles away from the water where you can work with them. We then bioassay every extract. A bioassay is simply a biological test. No matter what I'm studying, the biological assay controls the chemistry I'm going to be studying. If I'm testing for antibiotics, then I am only going to be isolating antibiotics because that's what my bioassay is going to lead me towards. So every organism produces compounds with an amazing array of activity, but it is our job to select what direction we go in. So we can look at enzyme inhibition assays or directly test against specific bacteria, fungi, viruses, and therefore direct our search. If we find something of interest, we then grow that same microbe in a much larger culture. We then go through a series of iterative chromatographic separations to isolate those compounds, always guided by the bioassay. So we specifically want to come up with a compound with an activity we care about at the end of the day. And then Don and I get to have the fun part of elucidating the structures using things like NMRs and mass spectrometers and infrared spectrophotometers to figure out what the structure is. Sometimes we'll do X-ray crystallography to determine the 3-dimensional structure because you need to know how that molecule fits into the enzyme where it acts. And then we'll actually isolate compounds that are closely related, just simply doing a different type of isolation because an inactive analog that's closely related can tell you something about structure, activity, relationships. And to a scientist, those are very important. So again, the bioassay directs the discovery. Our strategy, you need to select enzymes that you know are associated with a disease condition. You know you can inhibit that enzyme by particular chemicals, and that inhibition will block a reaction. You want to select an enzyme that is out of control in certain pathologies. You need to select an enzyme that can be inhibited, and after you validate your findings in these biochemical assays, you need to look at a cell system assay and finally validate those findings in animal models. And I'm not going to talk about any of this validation today. It's way too much into the weeds, but I am going to lead you through just a couple of the discoveries we've had looking at our enzyme-based assays.